This is a continuation of this morning lecture of the cell cycle. You have already started cell cycle this morning, so I will continue with the remaining part of mitosis and meiosis. So the objectives of this session, this lecture will be, the educational objectives are, at the end of this lesson, you are expected to list the phases of the cell, life cycle, the cell cycle, and describe the key events of each phase. You are also expected to describe the stages of mitosis. Similar, describe the stages of mitosis and identify cells at different stages of mitosis. And also you are going to describe the stages of meiosis. And finally, we will explain some clinical and medical aspects of mitosis and meiosis. Okay, the lecture outlines are, we are going to, in this lecture, we are going to cover the following topics. We will have an introduction, we will discuss about the significance of mitosis, and we will explain, describe the phases of cell cycle and the triggering factors for the cell to undergo cell division. And we are also going to we are going to discuss about the states of mitosis and the regulations of the cell cycle and the checkpoints of the cell cycle. The cell cycle is precisely regulated process, so we'll see some of the overview about the regulation and the checkpoints of the cell cycle and some clinical correlations. And finally, we'll also see the states of meiosis. Actually, the details of meiosis will be discussed in okay, the stages of meiosis will be discussed, the detail will be discussed in the gametogenesis. So mitosis, cell division are of two types in general. There are mitotic cell division and meiotic cell divisions. The mitotic cell division that occurs in somatic cells and resulting in the formation of two daughter cells which are genetically identical, and the other one is occurs in gametes in germ cells, and it is resulting in the formation of four genetically different gametes. Now, significance of cell division. Cell division in general is important for during the time of embryogenesis. As you can see, during the embryogenesis, cells will divide starting from the fertilized egg. The fertilized egg will multiply and it give rise to millions of cells, which these cells later differentiate and they will form the organs and systems of the body. When you come to mitosis, mitosis is a type of cell division in which the parent cell divides into two daughter cells, and the daughter cells are genetically identical. And the daughter cells, this is the cell cycle. Here is the parent cell. The parent cells divides into two daughter cells. The two daughter cells, again, they will grow and mature and attain the maturity of the parent cell, and the parent cell, and they also undergo another round of cell division. So this continuous cyclic process is called the cell cycle. So the cell cycle has different events in the cell cycle. In general, the cell cycle is divided into two. One is the mitosis, which is actual division that takes place during mitosis, and the remaining part is the interphase, which is a preparatory phase. In general, cell cycle, the mitosis has two major activities. One is the karyokinesis. Karyokinesis, it refers to nuclear division, whereas cytokinesis, it refers to cytoplasmic division. The karyokinesis has different stages: the prophase, promats. You can remember this by this the word: the prophase, metaphase, anaphase, and telophase. Cell cycle, as we have said, it is a continuous cyclic process or a series of changes of events a cell undergoes from the time it forms until it reproduces itself. That's called the cell cycle. The cell cycle is divided into two major periods. One is the interphase and the other is the mitotic phase. The interphase has different stages again: the G1 phase, the S phase, and the G2 phase. So this summarizes the stages of the interphase: G1 phase, the S phase, and the G2 phase. During the G1 phase, as you can see, these are the major events that occur during the G1 phase. The first one is, there is synthetic activity takes place during the G1 phase. The RNA synthesis of regulatory proteins, and synthesis of enzymes. These enzymes, regulatory proteins, and RNA are important for during the S phase, for the DNA replication phase. Okay, the DNA replication phase requires these regulatory proteins, these enzymes that are produced during the G1 phase. So the a pre-synthetic phase that prepares the cell to undergo the actual DNA synthesis. And as you can see also, the cell at the G1 phase, it will attain the size of the parent cell, and the nucleoli which disappear during the mitosis will be re-established. Centrioles start to form during the G1 phase. Centriole replication starts during the G1 phase, but it will be completed during the G2 phase. And another event during the G1 phase is centrioles began to the centrioles already, they start to replicate during the G1 phase and complete during the G2 phase. S phase is it is a phase at which the DNA synthesis takes place. The DNA replicates itself, and initially we have 46 chromosomes. In each 46 chromosome, each chromosome contains one DNA, but later the DNA will duplicate itself and become 4N. So doubling of the DNA. 46 DNA, now it becomes 72 DNA. So the main event activity that occurs during the S phase is DNA synthesis. The other one is the G2 phase. The G2 phase, again, it is a preparatory phase for the actual mitosis, where the spindle fibers form, RNA and proteins necessary for the formation of the spindle fiber are formed during the G2 phase. Energy will be accumulated during the G2 phase, and DNA replication is analyzed for possible errors, and these errors will be corrected during the G2 phase. So these are some summary of, you have already covered this in your previous lecture. This summarizes the different events that takes place at the interface. What triggers cells to enter the cell cycle? Cells do not randomly enter into cell division. There are some triggering events or events that stimulate the cell to enter into the cell cycle. The first one is a mechanical force, any stretching force of, like, for example, the smooth muscle, stretching of the smooth muscle, ischemia of the tissue, ischemia, cell death. These are the major triggering factors that allow the cell to enter into the cell cycle. So the cells do not randomly enter into the cell cycle. So there are triggering factors. At least you have to know these three important triggering factors. The role of these triggering factors is to release ligands, or these are chemicals known as the growth factors. These growth factors. The role of the growth factors is to stimulate the cells to initiate intracellular cascades. That these cascades will, finally, ultimately, will result in the expression of genes, proto-oncogenes. These proto-oncogenes are genes responsible for for cells to undergo division. So in general, the mechanism, in short, is first there is growth factor, which is produced as a result of these triggering factors. This growth factor binds to growth factor receptors, which are located on the cell membrane. Then these growth factors initiates cascades of intracellular signaling pathways. These signaling pathways ultimately will result in activation of transcription factors. These transcription factors, again, they will cause expression of proto-oncogenes. These are proto-oncogenes are genes in the chromosomes responsible for the cells, initiate the cells to enter into the cell cycle. So any damage to the proto-oncogenes result in uncontrolled proliferation of the cells during the cell cycle. So these mutated proto-oncogenes are known as oncogenes. So during the cell cycle, there are different types of cells. All cells do not undergo continuous cyclic process. Some cells, for example, they will undergo one or two turns of the cell cycle, and they will be arrested in the G of S, where they will perform the housekeeping activities, and when triggering factors are there, these arrested cells will enter back to the cell cycle. If need arises, these cells will enter into the cell cycle. But some cells continuously undergo cell cycle, like the epithelial cells in the GI, the skin cells continuously undergo the cell cycle without interruption, and these are called labial cells. Some cells leave the cell cycle and will be arrested in the geophase, and they will enter back to the cell cycle only stimulus. In the presence of appropriate stimulus, or in the presence of the triggering factor, the cells re-enter back into the cycle. These cells are said to be stable cells, like the liver cells, the pancreatic cells, the renal cells in the kidney are some examples. Some cells are permanently arrested in the geophase. They will not return back into the cell cycle. So they are permanently, these cells are incapable of undergoing mitosis. Like, for example, the matured nerve cells and the matured muscle cells, the skeletal muscle cells, the cardiac muscle cells. Once they are matured, they do not undergo mitosis. So these cells are permanently arrested in the geophase. So accordingly, we classify as labial cells, stable cells, and permanent cells, as we have described. So mitosis, it comprises two major events: karyokinesis, the nuclear division; cytokinesis is the cytoplasmic division. Karyokinesis is further divided into four distinct stages: the prophase, metaphase, and anaphase, telophase. So we are going to characterize each of these phases one by one. During prophase, from the G2 phase cells enter directly into the prophase. Prophase we can further classify as early prophase and late prophase. During the early prophase, the two major events are chromosome, chromosome condensation and the nucleolus disappearance of the nucleolus are the two major events that occur during the early prophase. As you can see, the nucleolus is already it disappears, no nucleolus, and the genetic material is condensed. Initially, the genetic material during the interphase is in the form of euchromatin and heterochromatin, as you know from your cytology class. But later, the chromatin material will be condensed and become the chromosomes, and with their sister chromatids. Okay, the sister chromatids, and found dispersed in the nucleus. Another event that occurs during the early prophase is the mitotic spindles will form, and the centrioles start to migrate to the opposite poles of the nucleus. These are the spindle fibers. They elongate, as you can see, they elongate, and the centrioles migrate towards the opposite pole. The other, so these are the major events that occur during the early prophase. So this is how the chromosomes get condensed during euchromatin and heterochromatin. First, there is DNA here. The DNA will undergo wrapping around the histone protein, octamer of histone protein, 1.5 turns, about 1.5 turns. This is the whole structure is called nucleosome, and with naked DNA in between. And these nucleosomes aggregate together and becoming the genetic material at this stage is called heterochromatin. And then the heterochromatin, it forms the loop like this. The loop will form further coiling and form this thick structure. So as the cell proceeds towards mitosis, as the cell approaches towards mitosis, then the genetic material, the chromatin, will become very condensed. It becomes very thick, and finally, as it enters into mitosis, it will become very thick and visible under light microscope with its sister chromatids. This is a sister chromatids. This is a condensation process, in short, how the genetic material gets condensed as it proceeds towards mitosis. Okay, so this during euchromatin, the cell is transcriptionally active, and transcription takes place for protein synthesis. And at that stage of heterochromatin, the cell is inactive, and it appears dark in the nucleus like this. So this is the heterochromatin. The lighter region in the nucleus is called euchromatin. So these two regions are metabolically, in the case of metabolically active cells, there is abundant euchromatin. In dormant cells, there is abundant heterochromatin. During mitosis, the cell will have these chromosomes. The same diagram. During the late prophase, again, sorry, as you can see, during the late prophase, to the opposite pole of the cell, the spindle fibers are elongated and start to attach. They are attached to the centromeres, attached to the centromere here, and the nuclear membrane start to disintegrate. So no more nucleus, the nuclear membrane disappear, and now the chromosomes are found being dispersed in the cytoplasm of the cell. And the mitotic spindles attached to the centromeres or to the kinetochores. So this is a major event that takes place during the late prophase. So the late prophase is characterized by disappearance of the nucleus, the nuclear membrane, attachment of the spindle fibers to the centromeres. Another phase of the cell cycle is mitosis, is the metaphase. The metaphase, it begins as newly duplicated chromosomes align themselves in the equator of the mitotic spindle. So the chromosomes with their sister chromatids are now aligned at the equator of the cell. See, this is the equator of the cell. This is when you see such kind of cell under a microscope, when the chromosomes are collected at the equator of the cell, then the cell is at the metaphase. The condensed chromosomes attached to the microtubules of the mitotic spindle at large electron-dense proteins. The specific areas are called the kinetochores. These electron-dense areas, which are located at the constricted region of each chromatid, called the centromere. These are the centromeres, areas where the two sister chromatids are attached, interconnected, is called the centromere. And there is an electron-dense area at the centromere, which is called the kinetochores. Anaphase is characterized by separation of the sister chromatids. As you can see, the sister chromatids now they separate and migrate to the opposite poles of the cell. The opposite poles of the cell, and they will be pulled towards the opposite poles of the cell. And during this time, the spindle poles also move farther apart. Some of the spindle fibers that do not attach to the centromeres will pull the two poles of the cell apart. And as a result, the cell size also increases during the anaphase. Okay, so this is the anaphase. So the major event during the anaphase is separation of the sister chromatids and move there to their opposite poles. This is a photomicrograph in your histology lab. You will see this kind of slides, this type of slides, and you are expected to identify cells under a microscope at various stages of mitosis. For example, this is anaphase. No, this is prophase. Prophase, metaphase. Anaphase, this is separation. The two sister chromatids are separated and they are migrating towards opposite poles. So this one set of chromosome, another set of chromosome, group of chromosomes are seen separately. So that is called the anaphase. Okay, anaphase. The previous one is metaphase. Finally, there is telophase. A telophase is the final stage of karyokinesis. During the telophase, as you can see, the sister chromatids now they occupy their location, the opposite pole of the cell, and the nucleolus reappear. The nucleolus will reform. The nuclear membrane start to form. The nuclear membrane will be formed, and so that is the telophase. So the telophase is characterized by the two sets of chromosomes at the spindle poles are begun to reverting to their decondensed state. Now the uncoiling process will take place. These chromosomes will uncoil and become changed into chromatids, and they will be stretched, become long and thin. So, and decondensed states, the chromosomes will change it into decondensed states. The microtubules of the spindles depolarize and the nuclear envelope began to reassemble around each set of the daughter chromosomes. And then cytokinesis, as you can see, cytokinesis is division of the cytoplasm of the cell after completing karyokinesis, nuclear division. The next will be the division of the cytoplasm. The cytoplasm start to divide, and this is through by forming constrictions. The cytoplasm will be constricted, the nuclear membrane will be constricted at the equator of the cell, and through these contractile proteins known as actin and myosin contractile proteins. These are contractile proteins aligned along the equator of the cell that cause constriction of the nuclear membrane. This nuclear membrane, the constriction, or it will become very deep, deepening of the furrows, and then finally the two cells will detach and form two separate cells. So that is called the cytokinesis. As you can see, here is a photomicrograph showing two separate cells, and here is a constriction of the nuclear the cell membrane, and then finally these two, we can see these two separate cells with their own genetic material. Okay, so this is about cytokinesis. So how this, as we have said, cell cycle is it's finely regulated process. Cells do not enter division randomly, as we have said, and the cell cycle is a well-regulated process. This regulation is through molecules or proteins known as intranuclear transcription factors. These intranuclear transcription factors, they regulate the expression of the proto-oncogenes. The proto-oncogenes then they allow the cell to proceed cell division. So the process, I'll give you the highlight how this cell cycle is regulated. Initially, there are cascades of intracellular signaling pathway. That signaling pathway ultimately it produces cyclins and cyclin-dependent cyclin-dependent kinases. Okay, this combination of these two molecules activates the intranuclear transcription factor. This intranuclear transcription factor regulates the expression of proto-oncogenes. This is D, okay, cyclin-dependent kinase, not G, cyclin-dependent kinases. These kinases and the cyclins combine together and forming, they activate the intranuclear transcription factor. The intranuclear transcription factor finally it regulates the expression of the proto-oncogenes. So the proto-oncogenes are one of the genes that regulate the cell cycle. So the capability of the cell to begin and advance through the cell cycle is governed by the presence and interaction of a group of related proteins known as cyclins and cyclin-dependent kinases. So there are different types of cyclin, D, cyclin A, cyclin B. So these cyclins regulate the different stages of the cell cycle. For example, cyclin D, it regulates the progression or the entry of the cell from the G1 phase to the S phase. G1 phase to the S phase is regulated by cyclin G, cyclin-dependent kinase 6, cyclin E, cyclin-dependent kinase 2. These three molecules, 1, 2, 3 molecules are important to regulate the cell to pass from the G1 phase, to proceed from the G1 phase to the S phase. So in the absence of these molecules, the cell cannot proceed from the G1 phase to the S phase. For the cell to proceed to grow from the S phase to the G2 phase, there is another molecule, cyclin, known as cyclin A. There is also cyclin B. This cyclin B complexed or combined with cyclin-dependent kinase 1 and permits the cell to proceed from the G2 phase to the M phase, to the mitotic phase. And there are also checkpoints. The cell cycle has two checkpoints. The first checkpoint is located here at the G1S interface, here, one checkpoint. Any error in the genetic material is checked at this point, the G1S checkpoint. Another checkpoint is between the G2 and mitosis. Okay, these are the two places in the cell cycle where the cell is monitored or checked for any genetic abnormality. If there is genetic abnormality detected, for example, at this area, at this site, then the cell will not proceed to the S phase. And instead, the cell cycle will be delayed in order to correct the error. The error will be corrected. If there is any DNA defect is detected, that defect or that abnormality will be corrected. Then after correction, the cell will go to the S phase. Without correcting the abnormality, the cell cannot proceed to the S phase. Another checkpoint is between the G2 and the mitosis. The similar activity also performed at this site. So these are the two checkpoints. The two checkpoints. What these checkpoints do, they detect any DNA abnormality. Any DNA abnormality. Okay, if the DNA abnormality can be caused by exposure of the DNA to certain chemical substance, exposure of the DNA to certain chemical substances, certain types of radiation, some viral infections can cause DNA damage. So a cell which contains damaged or defective DNA will not proceed the cell cycle, and it will be arrested at G1S interface, and the defect or the abnormality will be corrected, and then the cell will be allowed to proceed to the next phase. Otherwise, the cell will stay or at the checkpoint. So these are the main factors that cause DNA abnormality: chemical exposure, radiations, exposure of the cells to certain radiations, and certain viral infections will cause DNA damage. So a cell with DNA damage will not undergo cell cycle, and that abnormality has to be corrected first. So once problems are detected at the checkpoints, cells are arrested at G1 or G2 phase, then the cell cycle will be delayed in order to correct the abnormality. Then once the errors are corrected, then the cell proceeds to the next stage. This is what I've explained. What happens if the problem is not fixed or if the problem is not corrected? Then there is a gene called tumor suppressor gene. That tumor suppressor gene produce proteins. Those proteins may start to correct the abnormality. And the abnormality will be corrected by this protein, which is produced by tumor suppressor gene, as a protein such as p53. These proteins are activated, and the cell's activity is redirected towards cell suicide and apoptosis. So the tumor suppressor genes and the proteins that are produced by these genes will cause the cell to commit suicide. The cells will commit suicide and undergo apoptosis. Because these genetically abnormal cells by no means allowed to proceed the cell cycle, pass through the cell cycle. Because if these abnormalities are accumulated in the long run, the cell will become a cancer cell, and that cancer cell will later will multiply continuously and result in cancer or tumors. That is why the gene is called tumor suppressor gene. Tumor suppressor gene. So the tumor suppressor gene is a gene that produces a protein. That protein allows the cell to commit suicide on itself. Okay, and then it will be removed from the cycle. That abnormal cell will be removed from the cycle. What happens if the tumor suppressor gene undergo mutation? If the tumor suppressor gene undergo mutation, so the checkpoints will not function properly, and that abnormal cell will continue to pass through the cell cycle, and the abnormality will accumulate, and later the cancer will develop. So the gene encoding the p53 is often mutated in cancer cells, thus reducing the cell's ability to detect and repair damaged DNA. So inheritance of damaged DNA by daughter cells result in a greater frequency of mutations and general instability of the genome, which may contribute to the development of cancer. So this is how cancer develops in our body, mainly because of mutation of the tumor suppressor genes and the proto-oncogenes. So the two checkpoints are, these are the two checkpoints. Finally, we will see some of the clinical correlations of cell cycle. As we have said previously, cell to undergo cell division, there should be some kind of triggering factor. That triggering factor, as we have said, it is a growth factor. Growth factor. One of the well-known growth factor in our body is erythropoietin. Erythropoietin is a protein which is produced by the kidneys, and this protein stimulates the precursor cells in the bone marrow to undergo erythropoiesis. Erythropoiesis means formation of new red blood cells. So if there is deficiency of erythropoietin, as in the case of chronic diseases, and as in the case of kidney failure, chronic kidney disease, there is deficiency of erythropoietin, and as a result of deficiency of erythropoietin, erythropoiesis will be affected, and finally a person may develop anemia. Anemia is a shortage of red blood cells. So anemia, so in order to correct the anemia, sometimes it is possible to supplement erythropoietin. Erythropoietin supplementation can be given for the patient to replace the deficiency of erythropoietin. Another application of mitosis and meiosis, mitosis is the development of cancer. You can describe the pathogenesis of cancer by knowing these important genes, the tumor suppressor gene, and by the proto-oncogenes, the role of these genes. As we have said, the tumor suppressor gene, a gene that allows the cell to undergo apoptosis. If there is some defect in the genetic material, the cell will undergo apoptosis by itself. And that will be done by the tumor suppressor gene through its protein. By this, the cells can control the development of cancer. But what happens if this tumor suppressor gene is mutated? Then the cell will abnormally, continuously multiply, and forming a tumor. That tumor, it could be either benign or malignant tumor will result. Another proto-oncogene. Proto-oncogene is when it is mutated, it is called oncogene. So this proto-oncogene also result in the formation of cancer. I've already discussed this the previous session. Another important application of this topic is in cancer chemotherapy. Cancer chemotherapy means using of drugs to halt or to suppress the multiplication of the cancer cells. There are two commonly widely used drugs in chemotherapy. One is the methotrexate, the other one is 5-fluorouracil. Methotrexate is a drug that used to inhibit protein synthesis. Purine synthesis, which is part of the DNA. So it impairs the synthetic activity of the DNA. So it prevents cell multiplication. Another chemotherapy drug is 5-fluorouracil, which inhibits pyrimidine synthesis. Again, it is used in the during its effect is during the synthetic phase, during the S phase. So both of these drugs act during the S phase, and they are preventing cell division, and those are common chemotherapy agents. Okay. Okay. Finally, we will see meiosis. Hello, someone? Hello. Okay. Okay, good. Another type of cell division is meiosis. Actually, we'll discuss the detail about meiosis during gametogenesis, which will be your next topic in the next weeks. So this meiosis is a type of cell division which occurs during the germ cells. And it is a result in the formation of gametes. Gametes means spermatozoa and sperm cells, and the egg cells are called the gametes. Unlike that of mitosis, in the case of meiosis, the number of chromosomes are reduced by half. Okay, initially in the diploid cells, the 2n number of chromosomes will be reduced to n number. And at the end of meiosis, instead of forming 2 cells, 4 cells will be formed, and all of these 4 cells are genetically different. During meiosis, there are 2 crucial events or results. One is a reduction in the number of chromosomes from diploid state to haploid number. That is ensuring that each gamete carries haploid amount of DNA, that is 23 chromosomes, and haploid number of DNA. Another major difference from that of mitosis is there is a recombination of genes will recombine through 2 mechanisms. One is through crossing over, and the other is through reshuffling of the homologous chromosomes. These 2 activities result in the recombination of the genes. So the resulting cells are genetically variable and genetically different. So meiosis is, in general, is divided into 2 separate events, meiosis 1 and meiosis 2. Meiosis 2 is almost similar to that of mitosis, whereas meiosis is a reductional division. In this case, the homologous pairs of chromosomes, they line up. Homologous chromosomes means chromosomes having identical structure, and they are derived one chromosome from the father, another chromosome from the mother, are said to be homologous chromosomes. And these homologous chromosomes, they line up in the equator of the cell, and each pair separates. One homologous chromosome goes to one cell, and the other homologous chromosome goes to another cell. So each daughter cell receives half number of chromosomes, haploid number. Okay, meiosis two, in the case of meiosis two, it refers to separation of the chromatids. The chromatids, like that of mitosis, they will be separated, and one sister chromatid goes to one cell, another sister chromatid goes to another cell. So there is prophase one. During prophase one, you can see the homologous chromosomes, they pair each other. It's homologous chromosomes. And prophase one, okay. Okay, it begins after the DNA has been doubled after the S phase. Prophase one is a continuation of the S phase, and it lasts a long time and is subdivided into the following 5 phases: leptothene, zygothene, pagethene, diplothene, and diakinesis. These are different terminologies that are used to describe different events that takes place during prophase one. For example, during leptothene, the individual chromosomes become composed into chromatids, and these chromatids condense, form long strands in the nucleus. So this is the formation of chromatids and condensation of the chromatid. That's the first stage in the prophase one. The second one is in the zygothene phase. The homologous chromosomes, they will pair like this. They form tetrads. 1, 2, 3, 4 tetrad. In the pagethene phase, the homologous chromosomes, sister chromatids of adjacent homologous chromosomes, they will cross each other. And this crossing point is known as the chiasma. The chiasma, this is a site where exchange of the segment of chromosomes takes place. This is one cause of genetic variability. Crossing over is one of the genetic variability in meiosis. The other one is diplothene. Diplothene is chromosome continue to condense and then began to separate. They separate like this. They separate. And finally, dikinesis. Dikinesis refers to chromosomes condensation maximally, and the nucleus disappears, and as does the nuclear envelope also disappears, freeing the chromosomes into the cytoplasm. Now the chromosomes will be freed or dispersed in the cytoplasm of the cell. In the metaphase one, it is like metaphase of the mitosis. The chromosomes, the homologous chromosomes, they will align at the equator of the cell and forming the equatorial plate. In this case, unlike the mitosis, in this case it is the homologous chromosomes. Both homologous chromosomes align at the equator side by side. Okay? And during this phase, reshuffling takes place because one homologous chromosome, it occupies the upper part of the cell, another homologous chromosome to the lower part of the cell, and this alignment, this positioning of the chromosomes, homologous chromosomes, either to the upper part or to the lower part, is a random process. This random positioning of the chromosomes, homologous chromosomes, will allow reshuffling of chromosomes during metaphase. That is why all the resulting daughter cells have different homologous chromosomes So this is one source of variation in the case of meiosis. The other one is spindle fibers attached to the centromeres of the both homologous chromosomes. Anaphase 1 is evident when the homologous chromosomes pairs began to pull apart, they separate, okay, with their sister chromatids, they separate and migrate to the opposite poles of the cells. That is a major event during anaphase. So each chromosome still consists of their sister chromatids. The other one is telophase. During the telophase, the cells chromosomes, each consisting of two chromatids, reach opposite poles. After reaching the opposite poles, the cells will separate into two. So two cells will form, and these two cells again they undergo the second mitotic division, which is similar to that of mitosis. So each of these cells possesses 23 chromosomes. 26 chromosomes in this cell, another 23 chromosomes. Okay, so meiosis 1 results in the formation of two genetically different cells. These two genetically different cells then undergo meiosis 2. So in general, meiosis 2 is similar to that of mitosis. Here we have the two cells of meiosis 1. These two cells of the meiosis 1 with their sister chromatids, their chromosomes. This one's 23 chromosome, another 23 chromosome here, and these chromosomes will align in the equator of the cell. That is called metaphase 2. Metaphase 2. And in the anaphase 2, the cells separate at their centromere, and the sister chromatids migrate to the poles, the opposite poles of the cell. Okay, and finally, in the telophase 2, the cell, the chromosome will undergo uncoiling, decondensed. It will be changed into chromatin, and this nuclear membrane start to constrict. So that is the telophase. And finally, the two cells, the cytoplasm will divide through cytokinesis and resulting in the formation of four genetically different cells. So this summarizes the stages of meiosis 1 and meiosis 2. Okay, finally, we'll see some important clinical correlations of meiosis. This is abnormality in the chromosome number. As a result of abnormality of chromosome number, different types of disease conditions may occur. This abnormality of chromosome number is mainly due to non-disjunction of the homologous chromosomes. The homologous chromosomes may not separate, and the whole homologous chromosomes will migrate to one cell. As a result of that, the one cell may have 24 chromosomes, and the other cell may have 22 chromosomes. Because one set of homologous chromosomes, both homologous chromosomes may migrate to one cell, forming 24 and 22 chromosomes. One daughter cell with 24 chromosomes, another daughter cell with 22 chromosomes. If these cells fertilize, resulting in the formation of a zygote which contain 47 chromosomes. 47 chromosome is said to be trisomy. Another zygote will contain 45 chromosomes, which is monosomy. So non-disjunction commonly occurs in chromosome number 8, 9, 13, 18, and 21. Okay, we have 23 chromosomes. Each of these chromosomes are known by their designated by numbers. Chromosome number 1, 2, 3, up to 23. So commonly occurs in chromosome number 8, chromosome number 9, 30, 80, and 21. If non-disjunction in chromosome number 21, usually it produces a condition, a genetic syndrome known as Down syndrome. That is trisomy 21. Okay, trisomy 21. So during meiosis, one non-disjunction may occur. As a result of that, there may be unequal distribution of chromosomes to the parent, to the daughter cells. Okay, so this is all about our today's discussion. Okay, so thank you for attending this lecture. Okay, thank you. If you have comments and questions, you can inbox me. You can write in the comment section.